Hello, everyone. Welcome to this, the first CTSI Bird Seminar Series of this year, the spring 2025. Well, the fourth bird series is organized by Hershey Site of Bird. Bird is biostatistics, epidemiology, and research design core. And in the spring, the seminar series is organized by the UP site. And the co-host of this seminar series is myself, Mo-Sok Chow. I am research professor of statistics of the statistics department. And you can reach me at this email if you have any question about this talk. Prabhani is also co-host in future seminar talks, and she is also with the Department of Statistics. I would like to first show you the lineup for the bird seminars this semester. The first one is by Dr. Reinhardt. He's going to talk about functional factor analysis of brain connectivity. And we have one or more talks like each month in the following 4 months. In particular, you may browse through the previous year's seminars at the following site that I list at the bottom. Let me start with some Zoom webinar logistics. The Zoom section is being recorded. And please mute your microphone when our speaker is giving the lecture. Please feel free to submit questions using the chat box of Zoom during the lecture. You may use your microphone camera in the question and answer section at the end. And registrants will receive the slides, the recording link, and seminar feedback survey after the seminar. Well, let us start with today's talk. Today's talk is functional factor analysis of brain connectivity by Dr. Matthew Reinhardt, and he is principal research scientist at Amazon, and he is affiliate professor of statistics at Penn State University. I will do a short introduction to Dr. Reinhardt. He is a principal research scientist within the Amazon's reliability, maintenance, engineering organization. He got his PhD in statistics from U of Chicago, and he has a decade of experience in academia and industry. He leads teams in solving complex statistical, machine learning, and deep learning challenges. His expertise spans advanced forecasting, causal inference, and large-scale model deployment. His research interests include functional data analysis, longitudinal data analysis, and data privacy and application to omics data. And today, he's going to talk about this really interesting problem. Well, very difficult. Functional data analysis on brain connectivity. Well, Matthew, your turn.
Okay. Thank you very much for that nice introduction. Okay, so let's try and tackle the first technical hurdle. So I'm going to see if I can share my screen without everything breaking. How does that look?
Wonderful.
Okay, so
I can see, yeah, I can see what you share.
Okay, so I'm also going to try and go full screen and see if that breaks everything. Still good?
Looks great.
Okay, all right. Okay, so thank you very much. I don't actually give a ton of academic seminars anymore since joining Amazon. But when Mosek asked, I felt, okay, I should say yes. So I'll talk about some recent work here that Mosek has mentioned. So before going on, so I just want to emphasize that today I'm wearing my Penn State researcher hat and not my Amazon hat. I'm always happy to talk with folks about Amazon and my experiences working within Amazon. But today it's going to be mostly just Penn State professor work. So this is joint work with Kyle Stanley, who's a PhD candidate here in our department, in the Department of Statistics. And this is really his work. So I don't have really the bandwidth to kind of work on these sorts of complex problems without working with really talented students like Kyle. So this is really it's a joint work, but this is really coming from him. So this is a lot of the quality work. This is because of Kyle. And then so Nicole and I jointly advise Kyle, although I'm a bit informal because again I work now full-time at Amazon. Okay, so the presentation today is going to be fairly standard. So I'll kind of introduce the problem, talk about the model and the problems that we're facing. We'll talk about estimation, data analysis, and so on. So we'll sort of follow a typical structure. Now, if you're, I know most excited to save all the questions towards the end. If you're feeling really ambitious, I'm totally happy to take questions mid-talk. If I don't know if you're allowed to turn on your microphones, but if you wanna hazard it, I totally encourage it. I don't shove a ton of things into these talks to try and make sure there's enough time in case folks want to talk about something specific. So I encourage it if you can. Okay. So
Well, I think they can ask questions if they want.
okay.
I have not muted them, so
Okay,
they
good.
just
All
unmute
right.
themselves. But
Okay,
if
good.
you welcome questions, they can always unmute themselves and ask questions.
I'm more than happy if folks do it during the talk. It's totally okay.
Yeah, that would be wonderful.
Okay, so so this is going to be sort of my only explanation for fMRI and the underlying data. Really, Nicole and Kyle are much better versed than I am in terms of this type of data and these types of problems. So I'll give enough to sort of motivate the statistical work, which is where I really come in. And then we'll kind of revisit towards the end and sort of an application, looking at how these methods works with this kind of data. But for those that are unaware, just so we're on the same page, so when we're talking about fMRI, what we're typically talking about is these MRI images where we decompose the brain into these little things called voxels. And the voxels represent sort of how activated a particular region of the brain is by monitoring like how oxygenated the blood is. And so the data that we're going to be looking at is called resting state fMRI, which means that a subject goes into one of these machines and they just sit there. They're really told not to do anything, no task-specific actions from them. But a major motivation or a major question with this type of data is something called functional connectivity, which basically means how dependent are different regions of the brain upon each other, or do different regions of the brain kind of activate together or not? And how do they do it? So that's my fMRI slide. Okay, so but we'll jump more directly into the statistical models, because what we'd like to do is we like to be able to analyze data like this. And in particular, a big problem with these kinds of data is doing something like dimension reduction. You have so much data, how can we reduce it in a way that's manageable and understandable? So I want to start by talking about factor analysis. So for anybody that's had a class on multivariate statistics, hopefully you've encountered this tool and it won't look too crazy. But Melissa, can folks see like my cursor? I just want to make sure that if I point at something, you can actually see it.
What did you say?
Can you see my cursor? So if I'm like,
Yeah, I can see your cursor.
Okay.
Yes.
All right. So, okay. So that helps the discussion a little bit.
Right,
So
right. Yeah.
let's start by, so to give an overview of factor analysis, let's start by assuming we have this random vector. It's been centered, so we don't have to worry about that. And what we're going to assume is that this vector has a particular decomposition. And it decomposes into two pieces. So one piece is driven by what are called common factors. So you can think of these are factors, these are kind of underlying factors which are driving the dependence that you're seeing between the coordinates of x. And then you have sort of everything else that's left, which is considered just to be noise. Okay, and so you can write it down for each coordinate of this vector like this. If you prefer matrices, you can, or vectors, you can write like this. Okay, but you basically have this L, say, matrix, this matrix of loadings, and then you have all of these factors. And so the factors are random, the errors are random, and they're assumed to be independent of each other. The L, this loading matrix, so these loading values, these are parameters, okay, which have to be estimated, and they're common between subjects. So if I look at different subjects, I'll have different factors, different errors, but I'll have exactly the same loading matrix. So it's a parameter. And so what you typically assume with factor analysis, and a lot of this is to make sure that you can actually like estimate and identify everything, is that you assume that the factors, which are random, they're mean zero because everything's been centered. They're also independent or at least uncorrelated. So F1 and F2 or F1 and FK, they're uncorrelated. And then because there's a little bit, because you can sort of move constants around, so you can move a constant from L into F or F into L, we sort of assume that they also have constant variance or identity variance. And then probably the bigger assumption here is that the error terms are assumed to be also uncorrelated. So the error terms have, if we look at their covariance matrix, they have this nice diagonal covariance matrix. So it means the errors are not dependent between each other. And that is a pretty big assumption, actually, because what you're basically saying here is that with these factors, I've captured all of the dependence between the coordinates of x, and everything that's left is just kind of noise, just independent or at least uncorrelated noise. And so if this k, so if this is the number of factors that you have in the model, if this number of factors is a lot less than the dimension of the problem, then this is a real assumption. Like you're making a real assumption here. If k equals them, you're not making any sort of assumption. Okay. And so if you look at the covariance of x as a matrix, it means that it decomposes into these two pieces. So you have your loading matrix part, okay, which again describes all of the dependence between the coordinates, and then you just have this diagonal covariance matrix. Okay, and so this is the basic structure for factor analysis, the basic kind of model and underlying assumptions. Okay, so there is so what you typically learn when you learn about factor analysis is that there is an inherent problem here, or a potential opportunity, depending on how optimistic you are. So the problem is that the L matrix, it's not identifiable. So the way that this model has been written, it's not identifiable. So, and in particular, if I take an orthogonal matrix, R transpose, okay, so if R is an orthogonal matrix, which means that R transpose is the identity, what I can do is I can take this matrix and I can shove this R transpose in between the L's, right? So I can shove them in between the L's and I end up with this LR transpose, again, plus the D. And then I've rotated this L, so I get a different L, call it L star, right? But these two things are exactly the same, right? So this L transpose plus D, it equals L star transpose plus D, okay? And so in particular, if what that means in general is that this L is not identifiable. And if, for example, the Fs have like a spherical distribution, or just say like normally distributed, just to keep everything simple, then what it means is that L and L star, they'll lead to exactly the same data generating distribution, right? And if they lead to the same data generating distribution, then you cannot distinguish these two things based on data, okay? So that's a potential problem. This lack of identifiability, this is typically something that statisticians don't like, okay? But you can also view it as an opportunity in some sense, in that you can choose an L that you think makes more sense for the problem, right? So if there's a variety of L's which will sort of lead to the same data generation distribution, you can try and look for L's which have a particular structure, which is, say, easier to interpret, right? Or any other structure that you might like, right? And so a very popular approach to choosing the L is to use what's called a Varimax rotation. So with a Varimax rotation, you can think of it as trying to maximize the variance of the squared loadings, if you like. But there's a variety of different rotations you can also use. But the idea is that if you use something like a Varimax rotation, what's going to happen is that loadings are either going to be trying pushed up or pushed kind of all the way down to 0. So that's nice, because then what happens is that you'll end up with a loading matrix which is, say, fairly close to sparse, right? So you'll have some very high loadings and then lots of like 0 loadings. And then that usually means it's a little easier to interpret. So you can imagine if I come back here and I look at these loadings, say like the first factor, right, only has like 2 coordinates which it sort of loads onto. Then you can kind of group together those coordinates, and then you have some hope of actually interpreting them, because, oh, these 2 variables are grouped together. Are they sort of measuring or representing something similar or the same thing? Okay. So anyways, because of this, there was many years ago, there was a lot of different explorations about different types of rotations that one could use, which led to better structures. For this talk, I'll just focus on Varimax, but that's certainly not the only one. Okay. All right. So suppose that we were to try to apply factor analysis to fMRI. Okay. So here we have just kind of a simple example. So suppose that I have like a 5 by 5 by 5 voxel block, which is just a very small subset of the brain. And I wanted to use this data to do this. I wanted to use this data and apply factor analysis. Okay. So in that context, we'd sort of assume that we have x1 through xn from, say, different subjects, or it could even be the same subject, like at different times. And because we have 5 by 5 by 5, that means the dimension now becomes 125. So the dimension blows up on you quite quickly. And so if we just kind of ignore all the structure and we just feed it into a factor analysis model, what you'll end up with is sort of something like the following. So if you look at the diagonal matrix, this d hat, right? So this thing is supposed to be diagonal. If the model is correct, then really I should just be seeing a diagonal term. And this is a heat map of d hat. So I should just be seeing a diagonal term, and I really shouldn't be seeing these bands. So, and it turns out that it really doesn't matter how many factors you choose, like these bands are going to persist. So it would seem that it doesn't really matter how many dimensions you choose. You really can't get the factor analysis model to fit within this particular structure. And if you think about it, it's maybe not crazy, because if I try to put down this factor analysis model, there's going to be a huge amount of spatial dependence between these voxels. And the idea that you're going to completely capture it using this like simplistic structure is probably a little bit overambitious. So factor analysis applied to fMRI in general doesn't seem to meet the assumptions. Okay, so the goal of this work and of this talk then really is to sort of stop thinking about these things as just like vectors with unrelated coordinates, and try to put them in this paradigm called functional data analysis. Okay, where with functional data analysis, you kind of acknowledge that the data that you have, it's measured over some common domain. So the coordinates of x, they're actually tied together in some very particular way. They're not just anything. And so we can put all of this data and this problem within the context of functional data analysis, but let me give just a little bit of a backdrop of what that means. So sometimes called like first generation functional data analysis, it's sort of data that's observed over time. So that was where a lot of the initial work for functional data analysis was focused. It's where a lot of work still is focused. But the idea is that when I look at these coordinates, this, say, xim, so this is like the mth coordinate from the ith subject. When I look at this value, I'm acknowledging that this value is actually coming from some underlying function. Like there is some underlying function there that's giving rise to these particular values. And what happens is that I observe this function at, say, a particular time point. Okay. And now, potentially, depending on the problem, I might also observe a fair amount of measurement noise, and that measurement noise can be completely like IID, just completely random noise. But the idea is that there is something underlying these values, which is tying everything together, and it's tying everything together in some interesting way. So for example, like this function, maybe we think it's continuous, or it's differentiable, or it's L2 integrable, or whatever property you like. But we think there's this function has some underlying properties that we'd like to exploit. So when you view data like this, when you have this kind of multiple repeated functions, then this is, and you start developing statistical tools, this is an area called functional data analysis, and this is where the term comes from. My apologies to any folks in like bio or genetics who use functional in a different way. Unfortunately, there's going to be some overlap. So we can do something similar for fMRI. So with fMRI, it's almost exactly the same sort of message. It's just that now, instead of just having like, say, like a time point, we have a spatial location. So when we observe these voxels, the values of these voxels, we can say, okay, well, we think that this value that I'm observing is coming from some underlying spatial process, and then potentially maybe some measurement noise on top. Okay, let's see. I got a raised hand. Please.
Oh, hi, Matthew. Yeah, thanks. A very nice explanation of factor analysis and functional data. Just real quick, the fMRI brain volumes, are those the volumes of the more oxygenated blood?
Yes. So my understanding is, and if somebody has better fMRI experience than I do, then what we're looking for is activation, which means I think that the blood has been oxygenated.
Okay. So it's a measure of volume, and therefore it's reasonably continuous, and all
Maybe.
those other underlying assumptions or factor analysis of it, being normal and continuous and all that are
Right.
generally good. Okay.
I'll get to it. So I mean, you can disagree with some of these assumptions, but I'll get to more specific assumptions, actually.
Thank you.
So this is we're setting things up that, and maybe so that maybe in a way that makes sense. So okay, so as we said, so we think maybe that there is some underlying nice or nicer process describing the evolution of, say, these signals over the brain. Again, potentially with some measurement noise. Okay. And even if you don't think that things are necessarily continuous, then you might think, well, okay, but at least if points are very close together in the brain, then I might think that they're at least more dependent, right? And that this is something that I want to try and capture. Okay, so we'll see what we can do. Okay, so I'm going to present our kind of approach to this in two paradigms. Okay, so the first is going to be just assuming that all the data is completely observed. And by that, I mean, I'm going to assume that I have these underlying curves, okay, or these spatial maps, okay? And then the next paradigm will assume that, okay, well, suppose that that's not reasonable. Let's assume that we have something on a grid. And then what does the methodology look like then? Okay, but by understanding paradigm one, it can help us understand kind of the underlying structure and assumptions of everything. Okay, so all right, so how can we mimic the factor model structure for functional data? So this factor model is really set up for just an arbitrary random vector, okay? But if we know this data comes from functional data of some kind, as I've described, how can we start to mimic the structure of the factor model, or what changes do we need to make? Okay, so the most literal translation of the factor analysis model to functional data would look like the following. Instead of having coordinates of some vector, what you have is like spatial locations of some process, say over the brain or again over time, right? And so those loadings now become like spatial loadings. They become loadings that are across the brain or whatever domain you're interested in. But I still have these FK, these like one-dimensional factors, which again, the factors are specific to each individual person, but these LK maps, these spatial maps, these are shared amongst subjects. And that all seems fine. Like that doesn't seem to necessarily be a huge problem with this. And then we assume that there has to be some local error structure, right? So with the factor analysis model, you assume it's just completely uncorrelated, let's say. Okay. But the problem is that this assumption actually really doesn't make sense for functional data, right? To assume that actually, if I look at two spatial locations, then I'm going to end up with an error process that's completely uncorrelated. So we saw that with the plot, that in practice, you don't get that. But it just practically, that would just be an assumption that doesn't really make a lot of sense. And why would that not make a lot of sense? Well, you think that these x's have some underlying structure. Again, they could be continuous, or they could just be like square integrable or something like this. And if you put an assumption like this on top of your spatial process, it basically destroys all the structure. I mean, it's like adding like white noise on top of it, on top of your function. So you really don't have any nice properties of your function anymore. Okay, so this is a problematic assumption in general. Now, you could still assume that when I actually observe things, I get some extra measurement noise on top. That's all totally fine. But the underlying functions which are driving everything, it doesn't necessarily make a lot of sense to put down just a complete white noise on that process. Okay, so what might make more sense, or how can we sort of respect this local dependence structure without going all the way to this just completely like independence? Well, we can keep exactly the same factor structure. So this part was never really a problem. So the factors can be uncorrelated, mean zero, unit variance, it's all totally fine. The loading functions, we think they're trying to describe this kind of variation across the brain. So we think that it probably should be sufficiently smooth, okay? And maybe that's an additional assumption. But the assumption that we make instead of having this IID error or independent error across space is we just assume that we have a banded covariance. And so practically what that means is that I think that we're going to assume that the errors have a kind of a very local spatial covariance or spatial dependence. And as you move away, as points move away from each other, that this dependence dies off very, very quickly. Okay, and so this actually works very well with the fMRI connectivity sorts of assumptions, because when you're trying to do functional connectivity with fMRI, you might think that these distant parts of the brain, they're connected via this global piece, say these loadings, and that really everything else that you're observing is just, you still have this kind of local dependence, but that local dependence dies off very quickly, and it's not really the dependence that you're especially interested in, okay? So for scientific purposes, you might hope that a lot of the kind of interesting structure of the brain like gets captured in this first part, and this is just kind of a bit of this error is just a bit of a nuisance. Okay. All right. So if the global component of the covariance and the local component, so if we call the global component G and we call the local component B, just throw in some extra notation, then what this means is that if I look at the covariance of this spatial process, right, so this fMRI data, then what I think is this covariance is going to be split into two pieces. I'm going to have this piece which has all of those loadings in there, and they're going to describe some like lower dimensional, k-dimensional structure, and that structure is going to be kind of smooth, and it's going to describe kind of dependence between distant parts of the brain. And then I'm going to have this kind of banded covariance structure, which just describes like the local components. Okay. And if you really love math, then you can write this as covariance operators. All this. So when you deal with functions, the analog of a covariance matrix is a covariance operator. So if you like that sort of thing, you can write it like this, but it's by no means necessary. Okay. So, but now we come back to the identification problem. So, because the identification problem is potentially worse than with original factor analysis, because now what's happening is that we have this kind of structure. When we look at the covariance of our data, we have this banded piece where we observe both G plus B. So we have the global piece plus this banded piece. And then off of this band, I only observe G. Okay, but the thing is the only thing I really care about is G. So the question is, can I actually estimate this G even when I have this problem of having this contamination along the band of my covariance? And it turns out there's a very elegant solution coming from Victor Paneredos's group, which says that basically if you have, at least in the 1D framework, if you have analytic loadings, so that is to say if the loadings functions are sufficiently smooth, then it turns out actually you can estimate this G even when this thing has been contaminated by this banded covariance. Okay, so it's at least in the 1D framework, this was worked out that you could actually you could estimate these components, which is pretty cool. Okay, and so we worked it out that in the d-dimensional case that you have exactly the same property. It uses similar results. So this idea, if you have analytic functions, if you have smooth enough functions, then it turns out you do get this kind of uniqueness property, which basically says that if I look at two sets of global and banded covariances, and I see that they're equal, if those global covariances are really smooth enough, then the only way that the two things could be equal would be if each of the individual components are equal. So all this just says, basically the model is identifiable even when you have this banded contamination, which is nice. Okay, so now we can ask what happens when you go to the second paradigm where I actually observe things on a grid. Okay, because it's very nice if you have the entire functions, but practically that's not really feasible. So what we assume here is that we assume that again, imagine that we're dealing with voxel data or brain data. So we've taken this three-dimensional structure and we've cut it up into blocks. So in that sense, we assume that we have the X that we're observing on this each of these voxels. Okay. And so you can think that I end up with giant matrices or vectors or however you want to organize them. If you want to keep that sort of three-dimensional structure, then you would organize things as tensors. So instead of a two-dimensional matrix, you'd have, say, like a three-dimensional matrix or whatever the dimension is that you're working with. So we can evaluate this X on the grid. That gets us a tensor. We can do the same thing for the loadings. We can do the same thing for the errors. Okay, and so then in terms of models, what we end up with sort of like a tensor-looking model, and tensors are really cool, so why not reference tensors these days? So we have our observed tensor data. That is equal to some linear combination of these tensor loadings. Again, just think of the function evaluated on this grid, plus the errors. So exactly the same sorts of structure. Okay, and then if you think about covariances, so this is not super critical, but you can phrase everything in terms of, so if you're used to working with covariance matrices, you can phrase everything in terms of covariance tensors, and you get kind of like the same decomposition. So if I look at this covariance tensor, I get this one piece which describes all the all the global dependence between different parts of the brain. And then I get this banded piece. So again, all of this is just sort of thinking about the covariance, but now just evaluated on this gridded data. Okay. So not super critical. But one thing that is very interesting actually is that even in this context, even with data that's observed on the grid, as long as you observe enough data, you can actually still identify the parameters. So and by identify the parameters now, I mean the parameters on the grid. But as long as you have enough data, it turns out that this analytic assumption really does a lot of work for you. And so if you have enough data, if you have enough points, you can actually identify the model, identify all the components. And so in particular, this slide just discusses like how complex can the model be. So this K star, this represents like the largest number of factors that you could have in this model and have it still be identifiable based on the grid. Okay, the size of the grid. So for example, suppose that, so we're dealing with like fMRI data or something. So imagine that you had 100 points. So we're breaking things up into 100, we're breaking each axis into 100 pieces. So if I just have one dimension, then I just have 100 pieces, in which case the model can have up to 24 components. There can be 24 factors. If you have two dimensions, so you're thinking about like a matrix, so you have 100 by 100, and we have, 100 times 100 blocks. Then you can go up to over 500 components, and if you're up to 3, you can go up to 14,000 components. So you can have very large, complex models in these situations, and still be able to identify them, which is nice. Okay. So, and this is just formalizing this. So this is just formalizing that, yeah, if you, depending on how much data you have, as long as you have enough points in X, Y, Z, whatever directions you're looking at, the model should be identifiable as long as you don't have a crazy number of components, 14,000, you know, for 3 dimensions, let's say. Okay. All right. Okay. So, all right. So the estimation goal. Okay. So what now I've just been talking about the underlying model at this point and just trying to make sure that everything was set up nicely, everything is well defined, everything is identifiable. So now let's talk a bit about the estimation strategy. Okay. So suppose that we have just kind of a standard setup. We have IID data, and it follows some functional factor model with K factors. Okay. And we observe things on our spatial grid. Okay, and so again, now I'm setting everything up. So now I have one tensor, let's say, per subject, you know, which again is just think about their brain, the brain worth of their data. And so now I have the empirical covariance, which I can estimate. So I just can estimate all of the covariances between the different voxels using my subjects. Okay, that becomes the dimension starts blowing up on you very quickly, right? Because I have every possible combination of voxels. I can think about their covariance. But the goal is to estimate these loadings from this C hat n. And we don't want to use knowledge about, say, the factors or the bandwidth. We sort of want those things to be mostly data driven. Okay. Okay. So because I have forced so much math on you already, and I'm sure probably a lot of people are upset about that, I'm going to describe the rest of the estimation strategy via picture. So there are like loss functions you can imagine that are behind the scenes doing a lot of this work, but I think at this point, you know, we've all learned some pictures. So okay, so we are going to build our estimation strategy in a few steps. Okay, and so the first step is just an initial loading estimation. Okay, and this you can do with something like principal components. So you just assume that the loadings are orthogonal and get some initial estimation of these loading functions. Okay, and so the left here represents, so this is a simulation, but the left over here represents the truth. And then on the right, this represents our kind of initial estimate. And so on the right, you can see why folks, if you care about interpretability, then principal components is not always your friend, because the fact that things have to be forced to be orthogonal in this particular way, you end up with some really uninterpretable structures. And what started as kind of a nice, say, simple structure gets kind of destroyed by the principal components. Okay, but then after you do that, we use a rotation. So we rotate using Varimax. So we also incorporate smoothness at various steps, but let's just keep it simple. So we do a Varimax rotation. You can see with the Varimax rotation, you already are actually looking a whole lot better than the standard principal components. So you can see that you're now starting to really mimic the structure, but you get a lot of kind of little noisy bits around. And so then the third step that we use is just shrinkage. So if we have very small loadings, then we shrink them all the way down to 0 while trying to keep things reasonably smooth. Okay, and so what you end up with is you end up with something which now I think looks much closer to the truth and at least is certainly much easier to interpret. So you can see that the structure that we have in these 4 panels, you can sort of match in each of these examples, right? So this guy's over here, right, and so on. Okay. So we do initial loading estimation, we do rotation, and we do shrinkage. Okay, and then let's get this here. Okay. Okay. So so our major competitor is independent component analysis, or ICA, which is a very fun technique. So ICA is very similar to principal components, at least initially when you look at the actual structure, but the assumptions are quite different. So the assumptions for ICA are that your components that you have are basically independent of each other, and not too many of the components can be Gaussian. So I think you can have like one Gaussian component and that's it. The rest can't be Gaussian. But it turns out that if you assume that things are not Gaussian, then this is actually another strategy for solving this identifiability problem, or a identifiability problem. Okay. So what ends up happening is you end up with something which is a lot more interpretable than principal components, but you end up with something which is often very sensitive to the number of factors that you have in the model. Okay. And ICA, when applied to fMRI data, it still has exactly the same banding problem. So if you assume that you have like independent components or something like this, like some independent diagonal somewhere, it's a difficult assumption, I think, to believe in practice. Okay. So also, I mean, so just to kind of reiterate this point, so when people use ICA, they often like pre-smooth the data to get before you apply ICA to get better results. But again, if you think that things, if you believe in this, you know, if you don't want this kind of banded structure in there, or if you want this sort of diagonal, if you start smoothing things, you're going to start smearing dependence across the place. So this can make the problem worse. Okay. So we'll just compare to, so I'll give you some simulations and then we'll do an example. So we'll compare to a few different options. So we'll compare it to ICA. We'll compare it to ICA with smoothing because we wanted to see like how much does smoothing help you versus not help you. So it's because we have smoothing steps in our algorithm, but we wanted to see how big was the smoothing component. We also look at principal components. We look at the original method from Panaretos' group, which is a matrix completion approach followed by PCA. Then we have our approach, DPS, which is sort of, but it doesn't have the rotation and shrinkage piece. So you can think of it as having like this Panaretos approach, but with smoothing. And then you have our full thing. So you have our functional FFA model. So it's the full smoothing, rotation, shrinkage. It has all of the pieces all combined. Okay. So we just wanted to do this in stages just to see like what is contributing where. Okay, so so here are the simulation parameters in case you care to know. So we have similar structure before. We'll do different numbers of Ks and bands and sample sizes and so on. Okay. So hopefully my screen's pretty big, so hopefully you can see this. If you can't, then I apologize. So if we compare sort of all of the different methods, so our approach is going to be the baseline. So ours is sort of going to be a 1, and then everything else is going to be kind of a ratio of how much worse it is than our approach, right? So if it's 2, then it's the errors are twice as bad in terms of estimating these components, okay? And so what you see is that it doesn't really matter what the parameters are. Our approach seems to do a pretty good job. We beat all these other competitors. Of course, we're designed for this, so that's not too surprising. One thing that is sort of interesting is, so if we say focus in on like the red and this kind of greenish color, this, I don't know, forest, I don't know what you call this, but if say like this pair and this pair and this pair, what you'll see is that the smoothing really does a lot of work for you. So the smoothing really does help you in terms of getting better parameter estimates. And this is sort of true across the board. So if you look at the other pair of ICA and ICAS, so this teal thing is ICA, and then the blue one is going to be ICA with smoothing. You see that it sort of always helps you. So smoothing is really, it seems to be that smoothing really makes a lot of sense for this type of data, or again, in these simulations. But a big amount is just getting the smoothing. So not even just the rotations and things, but even the smoothing is still helping you quite a bit. Okay. All right, and then I think this is fine. I mean, but these simulations are designed for our model, so which is how it always is. But I think the more interesting thing is the interpretability of the results. Okay, so here's an example where we go all the way up to say 8 components, so 8 factors. Okay, and then we use different approaches for estimating them, right? So this is the truth, and then here's our approach for estimating these different components, and you see we do a pretty good job. Like some of the later ones get a little bit weak in terms of the signal, but everything else looks pretty good. If you use ICA with smoothing, it's okay for some bits, and then say for a few components, and then you basically kind of lose it later on. And then if you do this DPS, which again, you can sort of think of it like principal components, you kind of don't recover any of the structure at all, right? Everything gets kind of destroyed. So you also end up with something which is much more interpretable if you do our approach. Okay. And the other thing is this sensitivity for ICA. So ICA is an interesting method, but it is quite sensitive to the number of components. And in particular, what happens, this just sort of illustrates that if you take too many components for ICA, like so you have this kind of nice structure that you're estimating with ICA, except maybe kind of miss it at the end. But if you start adding in too many components, what will happen is the ICA will start splitting them into pieces. In which case, maybe I don't know, the first two guys are okay, but everything else has been kind of like split up. And so this is kind of a known weakness of ICA. There are approaches to try and fix this, but it is kind of something which our approach doesn't have a problem with. So if you look at our approach, if you just keep plotting these extra guys, what will happen is will just kind of be shrinking further and further down to 0. And you'll see that basically after 8, you're not getting really any additional components that are interesting. Okay, so all right, I'm getting a little bit low on time, so I don't want to get too much into the details of how things are set up. We use lots of standard pre-whitening and pre-processing of the data that is applied to fMRI, so none of this is novel on our part. And for visualization purposes, we're just going to look at one like horizontal cross-section for a subject, and we're going to look at them over time. So our repetition is going to be like over time. We're taking multiple scans of the subject over time. Okay. And so if we do, in this one example, so this is now applied to real data. So again, I don't want to really stand here and claim that like our structure is better than ICA, because that involves much further arguments. But it's just to highlight some of the splitting that happens. So if you look at sort of our structure with different numbers of components, so this is the 5th component but we fit different size models, so either 12, 25, 50, or so on. And you see that every time it basically comes out to be the same thing. I mean, there's like a little bit of variation, but it's not super big. Whereas again with ICA, so over here we have the ICA approaches, which end up basically splitting things into pieces. So you have one big structure, and like for a certain number, certain K, it keeps that structure, and then subsequently it ends up tearing it apart, which I think is maybe not necessarily the thing that you always want to do. Okay. All right. So yeah. Okay. So with that, so I just want to mention that putting fMRI aside, principal components has been like the dominant tool within functional data analysis. There's practical reasons for that, and there's mathematical reasons for that. But I think maybe it's probably should be expanded a bit, and factor analysis is maybe one approach if interpretability is especially important. If all you care about is sort of dimension reduction for estimating your model, some subsequent model, sometimes it's not clear that you need to move away from principal components. But if you really care about interpretability, then I think principal components is kind of not a great choice. And so now swinging back to fMRI, I mean, ICA is the heavily used approach, but it seems like maybe there's some potential other options, especially if you start thinking about the underlying assumptions that need to be made for these things to work properly or guarantee that they're working properly. ICA, I think, maybe has some questionable assumptions. We have our own set of assumptions, so you can look and see what you prefer. But in general, I think this is kind of an interesting area. fMRI combined with functional data. I think this is a really interesting area because you get a lot of really complex data with a lot of complex structure, and you can build very interesting models. And really, I've only talked about the spatial component, but with fMRI studies, you can have both a spatial and a temporal component, which I think opens up the complexity even more and opens up for some fun models. So with that, thank you very much. And if you have questions, I'm happy to chat.
Matthew, thank you very much. Great talk. I wondered, a lot of folks who use factor analysis, sociologists and such, they've really embraced not the orthogonal, but the other set of rotations. What do
Oh,
you
like
call
the oblique rotations.
Yeah, the
and
oblique rotations. Do you find that's true also, that they claim that, in fact, right now I took a course in this last year, and the sociologists were saying that they actually distrust any factor analysis solution that doesn't also examine the oblique rotations.
Sure.
How much does that complicate things in your scheme?
I don't think it complicates things very much. So if you want to use different types of rotations and you want to allow for these oblique rotations, there's nothing inherent about the structure of what I described which precludes it. So you certainly can do it. I think that it is actually an interesting point, because I think that there are so with Kyle and with Nicole, like we've been recently looking at both space and time as components in some of these models. And there, I think there is some evidence that relaxing this kind of orthogonal structure might actually be very advantageous, in the sense that depending on what you want to assume over space and time, in order to keep this kind of identity structure for the factors and just use these orthogonal rotations, like you have to make some pretty strong assumptions. So I think it's a good direction to go in. And I think that, at least for certain applications, I think probably it makes a lot of sense that the assumptions are more realistic. So I'm certainly encouraging. And again, if you want to use there's really nothing here that's super special about you have to use this rotation or you have to assume this type of structure. Like it's a fairly open framework in that sense.
Hi, Matt. There is a question on the chat. How
Oh,
does
okay.
FDA relate to state space models?
How does FDA relate to state space models? I don't know. I don't study state space models very closely, so I don't know if I can give you some deep, interesting answer to that question. So I probably will just punt it. So, but let me just say what are you trying to do with functional data analysis? So with functional data analysis, you're trying to keep things fairly non-parametric. So that is to say, when you study processes, you maybe assume that they have some amount of smoothness, right, or continuity or something like this. But beyond that, you really don't want to impose any additional structure, or certainly parametric structure. So it's really something which is sort of coming out of non-parametrics. And so in that sense, you're really trying to keep things very light and very general, and you're just trying to exploit some very general structures that you believe are in the data, like I said, like smoothness. And the reason that you can do this is because you have repetition. So unlike time series models or classic time series models, where you might only have one time series and you have to make do with one time series, so you need a very simple model to model a single time series. With functional data, you have repetition. So you have repeated versions of your time series or your stochastic process. So it means that you can make a lot less assumptions if you want.
Yeah, hey, Dr. Reimers. Thanks for
Alan,
So
how's it
I
going?
do have one question. So here is that I think one interesting thing is that you said that with increasing dimensionality, you actually can allow for an exponential growth of components considered, right?
Yeah.
So my question is that since in such a case you are considering my understanding is an additional correlation structure. So we are considering such an additional correlation structure will also lead to somewhat like an exponential growth in computation with respect to the dimensionality.
Yes, 100%. So the computation, so the biggest struggle I think with these kinds of models is the computational complexity. So the moment that you go into 3 dimensions, the size of the data, the scale of the computations, it grows very rapidly, to the point where you have to be very careful about how you compute things. So you can't just throw things into R or Python or whatever, and just
right,
hope for the
that makes
best. You
total sense.
you have to be very careful with the algorithms you use. And so there are different approaches you can take, and Kyle knows these much better than I do. But I think there are certain approaches which allow you to update. So probably one of the worst things that you have to do is like if you have to estimate the full covariance and then store it in memory and then work with that thing, that's terrible. So anything you can do to avoid that. And there are certain tricks that you can use to like update parts of the loading tensors or functions or whatever you want to call them, like with subsets of data and things, and try and avoid that. But it's a major problem, and I think there's some interesting research opportunities there in terms of understanding how do you exploit this structure in a way where you can compute things but avoid some of the computational headaches.
Yes, that makes total sense. Thank you.
Thanks.
So, Matt, can you give the audience your email so that if they have questions, they can email you?
Yeah, I will. How about I give you my email and you can send it to the audience, and then
Yeah.
and then everybody will have it if they need it.
Yeah. So this is great.
Okay.
And we are
Thank you so
Thank
much.
you. Yeah, thank you so very much for your talk, for giving such an interesting talk. And it is now 2 p.m. So thanks. And if there's questions, we are going to send you the email. Okay.
That's good.
Dr. Rama's email.
I'm also, I mean, it's not too difficult to find either. So it's
Right. Yeah, true.
But I can send it out.
Okay. Thank you.
Thanks all. Bye.
Bye-bye.